Kita lanjutkan selanjutnya dengan pembicara ketiga, yaitu Profesor Lawrence Yang. Sebelumnya, seperti yang lain-lain, akan saya share Profesor Lawrence Yang, Ph.D. Beliau saat ini, sebentar, saya lihat ke PA-nya ya. Beliau saat ini menjabat sebagai co-lead ya di University di New Warwick Health Global Research Priority, and beliau adalah staff di Medical School University of Warwick. Beliau pernah menjabat sebagai Kepala Divisi Studi Kanker di University of Birmingham pada tahun 2001 sampai dengan 2007. Kemudian juga pernah menjadi Prof Vice Chancellor di College of Medical and Dental Sciences di University of Birmingham pada tahun 2008. Kemudian menjadi Vice Rector di University of Warwick, United Kingdom pada tahun 2013, yang bertanggung jawab pada strategi perencanaan dan juga sumber daya. Kemudian pernah menjadi wakil rektor di University of Warwick dan bertanggung jawab pada kolaborasi dengan university di China dan juga Hong Kong. Beliau juga sebagai professorship kehormatan di University of Birmingham, United Kingdom, kemudian sebagai profesor kehormatan di Monash University, Melbourne, Australia, kemudian visiting professor di Cancer Center Sun Yat-sen University, Guangzhou, China, dan juga di Fakultas Kedokteran, Kesehatan Masyarakat, dan Keperawatan UGM. Bidang riset beliau yaitu peran infeksi virus ya, terutama Epstein-Barr virus dan juga human papilloma virus pada pathogenesis berbagai tumor. Kemudian beliau juga banyak melakukan penelitian pada terapi gen dan juga imunoterapi yang kemudian diterapkan pada berbagai uji klinis. Beliau mempunyai banyak sekali publikasi, lebih dari 230 publikasi. Beliau juga mempunyai banyak sekali kolaborasi baik yang bersifat nasional maupun internasional. Okay. Good morning, Lawrence.
Hello, hi.
Hi. We are very happy to have you.
Lovely to be here. I'm going to try and share. You'll have to let me know that if it's working okay. Okay, is that all? Does that look okay?
Yes, Lawrence. Yes, you can start.
Okay, thanks. Well, thank you very much for the opportunity. I'm really pleased to be talking about my favorite subject, which is the association of infections with cancer. I'll briefly introduce the field and talk a little bit about some of the numbers, some of the epidemiology, before focusing on two of my favorite viruses.
Sorry, interrupt, Lawrence. Your time is 35 minutes, okay?
Okay, I shall speed up.
Thank you.
Okay. So what we know of cancer, really, one level is quite simple, another level is quite complicated. The simple bit is that this is a disease where environmental factors impact on our DNA in some way. And we're all familiar with the different factors that affect cancer. That is dietary factors. There are also factors associated with, wait a minute, something's not happening here. So the dietary factors, smoking, sunshine, we know UV light is very important. All of this is about the way that DNA is affected by environmental factors and by mutagenesis, by changes in the DNA. But on the bottom left here, you can see another important contribution, which is the contribution of infection, and how infection, through different mechanisms, can impact on DNA and contribute towards the development of cancer. Every so often, there are these large epidemiological studies of cancer around the world. One has been published more recently in 2018. This is just data from 2012, but the data is more or less the same. It demonstrates that around 15% of all cancers have some relationship with infection. If you look at the list here, you can see at the very top, the most common form of infection associated with cancer, in this case, stomach cancer, is infection with a bacteria, Helicobacter pylori. But as you go down the list, you'll see that the majority of infections associated with cancer are due to virus infections. Another interesting fact associated with this epidemiology is when you look at the overall geographical variation in the association of infection with cancer, what you find, and you can see it here highlighted at the bottom, is that for some reason, which is still difficult to explain, we still don't fully understand this, the majority of infection-associated cancer is found in less developed regions. You can see here that's 23.4% as opposed to 9.2%. So there is something going on in less developed countries that seems to drive a higher prevalence of infection-related cancer. The list here is an up-to-date list in order of the prevalence of different cancers and their association with different viruses. So you can see that the top of the list is liver cancer associated with hepatitis B and C viruses, cervical cancer and other forms of anal genital cancer with human papillomavirus. Something I'm going to talk about, which is the association of a number of different tumors with Epstein-Barr virus. The only retrovirus in this list is the human T lymphotropic virus, which is associated with adult T-cell lymphoma. Kaposi sarcoma is a herpes virus. Merkel cell carcinoma is a polyoma virus. So these are different viruses. The majority of these viruses are DNA viruses. What that means is their genome, the viral genome is DNA. Now, what we've learned for many, many years studying virus-associated cancer is a few key criteria for associating virus infection with cancer, and a few headline issues around what the viruses are contributing to cancer development. So here we have the characteristics of an oncogenic virus, a cancer-causing virus, and these are modified versions, if you like, of the postulates for linking an infectious disease, an infection to a particular disease. So what you'd expect if you were trying to attribute a virus somewhere in the etiology of a cancer is that the individual with the tumor would have some evidence of prior infection, and the way that we tend to measure virus infection in individuals is antibody responses in the blood. You'd also expect that if the virus was in some way contributing essentially to the development of a tumor, that you would see the virus or bits of the virus in the tumor itself. So you'd expect to see the viral genome, be it DNA or RNA, in the tumor, and you'd expect to see RNA and proteins that are being made by the virus and in some way contributing to the development and maintenance of the tumor. And you might even, under certain situations, be able to identify infectious virus from that particular tumor. These studies are often helped by being able to develop an in vitro model. And so you might expect that either the virus itself or viral genes could be used in some way to contribute in an in vitro test to transformation, to cancer, like phenotype in tissue culture. And you might even be able to have an animal model that helps you study that in more detail. These are still very difficult criteria. And the ultimate proof really comes from being able to, in some way, intervene in the process of cancer development with a vaccine or a drug that blocks the virus. What we have learned, however, is like all cancer, it's not just one thing, and the virus is just one factor. What tells you that, what supports that, is the long latency period between primary infection and tumor development. So you might get an infection when you're very, very young, but not develop the cancer until 50 years later. And what that tells you is that it's not just the virus infection itself, but other factors working together with the virus that contributes to cancer. This is also supported by the fact that only a small percentage of people infected with the virus will develop the tumor Again, evidence that the virus infection is just one step in a very complex multi-step pathogenesis. The virus is just one link in the chain, but our belief is if you can break that link, then you'll be able to affect or stop the development of cancer. There's a long history through last century, through the 1900s, of the association of virus infection with cancer in animals. And you can see these slides later, and I won't go through this in any detail, but this history started in the early 1900s. And it wasn't until 1964 that the first virus associated with human cancer was discovered. And that is the virus called Epstein-Barr virus, named after the two people who were principally responsible for discovering this virus. And so it wasn't until 1964 that all the work that had gone on before in animals became relevant to humans. And I'll mention that a bit more later. And there's some other key milestones here that that you can look at afterwards. So if we come on to Epstein-Barr virus, we go back to the epidemiology. What you can see here is that Epstein-Barr virus is associated with probably something like just over 200,000 cases of cancer a year. And we know from work that goes back many years that this is probably a significant underestimate because of the difficulty in certain countries in terms of cancer registration and the prevalence of Epstein-Barr virus associated cancer in parts of the world where collecting epidemiological data can sometimes be more challenging. What's interesting, of course, coming back to what I said earlier about infection-related cancer more generally, is that when you look at these EBV-associated tumors, they are predominantly in East Asia and Southeast Asia, significant numbers, and that's because of the particular association of Epstein-Barr virus with a tumor that you're going to hear a little bit more about shortly, called nasopharyngeal carcinoma. So here again, something you can look at in more detail later, a detailed timeline of the development of our understanding of Epstein-Barr virus and its association with cancer, and many of the key developments over many, many years. What's an interesting one here is in 1984, when the EBV genome was fully sequenced. That at the time in 1984, that was the largest piece of DNA ever sequenced. The EBV genome is 172 kilobases of DNA, and it's almost laughable now that we're in an age where we can so easily sequence the human genome. But in 1984, the year I actually joined the field working on EBV myself, the virus was fully sequenced, and that was considered to be a very significant breakthrough, being able to fully sequence 172 kilobases of DNA. So Epstein-Barr virus is interesting because, quite surprisingly, once it was discovered associated with a particular tumor called Burkitt lymphoma in Africa, it was then subsequently shown, firstly by serological assays, that this virus is a very common virus. In fact, Epstein-Barr virus remains the most common virus infection in humans across the whole world. Most people, about 96% of the world's population, are infected and carry this virus. It's a herpes virus, and we carry it in a lifelong carrier state where we have no symptoms. We get infected when we're young. It stays in our bodies as a persistent infection, very similar to other herpes viruses, and very rarely does it cause any concerns for us. So this is quite interesting and also quite frightening that we're all carrying a virus associated with cancer, but that it's kept under control, mainly by a very strong immune response in our bodies. And the virus, like other herpes viruses, displays a dual tropism. That means it can infect two different tissues. It infects our B lymphocytes, where the virus actually persists. So the long-lived home, if you like, of EBV in all of us who are infected are memory B cells. But the virus can also replicate in those B cells, but more often replicates in the epithelial cells in our nasopharynx. So we'll hear more about that shortly. So here's an interesting thing. This is a very common infection. And this is also true of most of the other virus-associated tumors, that the viruses are very common infections. EBV, however, is associated with two broad categories of cancer. The cancers occurring in immunosuppressed individuals and cancers occurring in the immunocompetent. In terms of immunosuppression, it's not it's common that virus-associated malignancies, which are very strictly controlled by our immune system, are more prevalent in individuals who are immune suppressed, because of course that immune suppression removes the ability of the immune system to control the outgrowth of those tumors. And EBV is a very good example of that, particularly with B-cell tumors, both in post-transplant patients, people who are being immune suppressed to prevent the rejecting a transplanted organ or transplanted stem cells, or indeed originally when we and others started to look at HIV, we found that HIV is obviously is associated with lymphomas. And again, all of this is a consequence of the removal of the immune control over EBV, and this is predominantly an effect on B cells resulting in lymphoma. But then there are these malignancies that occur in immunocompetent hosts, which are a bit more difficult to understand and a bit more complex in their etiology. And those are tumors like Burkitt lymphoma, where the virus was originally identified, Hodgkin's lymphoma, a range of T-cell tumors, and then a couple of tumors that are not in the lymphocyte lineage, but are in epithelial cells, nasopharyngeal carcinoma and gastric cancer. Now, EBV is a really interesting virus because it is the most efficient virus at transforming human cells in culture. And so if you take B lymphocytes from your blood and infect them in culture with Epstein-Barr virus, around 30% of the cells will become transformed. They become immortalized and will continue to grow in tissue culture as cell lines. This is a very important tool in many respects for many different studies, actually, not only in EBV, but also in sort of rescuing the genome. If you want to do genomic studies where people have done work, for instance, on mismatch repair defects in genetically in individuals, you can use this type of system to rescue the genome. What's interesting about this is that this virus, which has 172 kilobases of DNA, can encode more than 100 proteins, actually just uses a handful of proteins and genes to elicit this effect in culture, which is a very powerful effect. And it's quite an interesting one. And you can see highlighted here are really three groups of genes that the virus encodes that contribute to this effect in vitro, this powerful effect. They are nuclear antigens called the Epstein-Barr virus nuclear antigens, the EBNAs, that help in some way in controlling gene transcription. But one of them, the EBNA1 protein, is really important because that helps to maintain the viral genome. This virus doesn't integrate into the host chromosome. It stays as extra chromosomal circles of DNA, and those circles are maintained by the EBNA1 protein. There are two really important membrane proteins associated with transformation. One of them is the major transforming protein called LMP1, and the other is LMP2. They're both at the surface of the cell, and they are important in cell signaling and infecting cell signals. And then there are a group of RNAs encoded by the virus, some non-polyadenylated RNAs called EBAs, which seem to be important in immune control, and then a range of microRNAs, including something called the BART. So this is the virus in which microRNAs were first discovered, actually. And the virus uses these, again, as a way of regulating viral gene expression. Here's what we think is going on. So cutting a long story short, what we believe is going on here is that this virus has evolved to enter and stay latent and persistent in memory B cells. And it does that by essentially overtaking and controlling the development of B cells. We know that B lymphocyte development, so that cells can become antibody producing cells, takes place in germinal centers. And what this virus has done, it has essentially parasitized that process. It's learned over evolutionary time how B cell differentiation works, and it uses various genes and various patterns of transcription to overcome and drive the development of memory B cells in predominantly an antigen independent way. So whereas this process is usually governed by B cells seeing antigen, that process is usurped by EBV. And this little cartoon summarizes some of that and demonstrates that some of the cancers that we see from the B cell lineage either developed from naive B cells or from germinal center B cells. That's a bit more detailed, but you can also see that this is a virus. If you look at the left-hand side, that the B cells can produce virus, and the epithelial cells can also produce virus. And it's in that context that we know the virus contributes somehow to the development of this tumor called nasopharyngeal carcinoma. So it's a complex process, but one where the virus has very cleverly developed a mechanism for persisting in memory B cells and living with us in a way that we don't even know. So most of us don't even know we've got this virus. So as I mentioned earlier, the virus is a circular genome in infected cells. It circularizes 172 kilobases of DNA. At the bottom here is a linear version. So the virus is double-stranded linear DNA within the virion, and it encodes all these loads of different proteins. And we know that as the virus replicates, it uses up to 100 different proteins to replicate itself. But this is very, very strictly controlled. And this whole transcription regulation results in different forms of virus gene expression that is referred to here as different forms of latency. And again, I won't go into details here, but these are different stages of gene transcription on the viral genome that are used mainly to drive the virus into B cell memory. This can all go wrong, and what happens is these latency forms can be frozen in different cancers, and we are still grappling with how all of that works, knowing increasingly that not only these latent proteins, but also some of the proteins involved in virus replication can contribute to cancer. But simplistically, what we've got here is a situation where the virus replicates in epithelial cells in our nasopharynx, in our mouths, throats, and noses. The B cells can become infected and become memory B cells. But occasionally what happens in our bodies, all of us are infected, is some cells will become like those cells that you can transform in culture when you infect B cells with EBV. They'll become transformed in our bodies. But that process is very strictly controlled by a strong T cell response, predominantly a cytotoxic CD8 T cell response. So this immediately tells you that anything that stops this T cell response from working effectively will allow these cells to grow out and not to be controlled. And that's exactly what happens in these types of tumors that you get in immunosuppressed individuals. These tumors look very much like the cells that you can develop in culture when you transform B cells. These lines are called lymphoblastoid cell lines, or LCLs, and these tumors in humans who are immunosuppressed look like this. And what you can see is, in a really interesting approach that tells us a lot about cancer generally, is the development of monoclonal non-Hodgkin's lymphomas from polyclonal diseases. So you get a lymphoproliferative disease where the B cells are proliferating. As they proliferate, they accumulate other genetic changes, and they then move from polyclonal proliferations to monoclonal tumors. But of course, the reason for mentioning this is that you can treat these with adoptive T cells. So you can simply put these cells back. You can grow from the blood of infected individuals, that's most of us, specific T cells that will target EBV, and you can inject those back into people, and you can actually prevent and indeed cure both lymphoproliferative disease and certain B cell tumors. So this is the first and most direct example of using adoptive immunotherapy to treat cancer. EBV is the paradigm. It's the model for that type of therapy that's now being explored in lots of different types of tumors. Now, quickly coming on to nasopharyngeal carcinoma, because of the interest, this is a common tumor in Southeast Asia. You can see the world distribution here. In certain populations, you can get up to 30 cases per 100,000 per year. It's a complex interplay between environmental factors and genetic factors and EBV. So there are a number of factors, and we know that one of them I know that's been worked on by Yaya is the association of environmental factors like salted fish in this development. So again, this tells us that even though we know, as you'll see in a minute, that this tumor is associated with Epstein-Barr virus, it's not just the virus, it's other factors. And that's what fascinates us about this unique tumor, It occurs at a particular part of the upper nasopharynx where the eustachian tube from your ear meets the upper nasopharynx, a very specific area. It has a very significant histology that is very much dominated by stromal cells and infiltrating lymphocytes, which makes it a really interesting tumor to study when we're looking at this very topical area of the tumor microenvironment. We know it's a combination of environmental factors, but EBV is there all the time. And because it's there all the time and is important for continuing and maintaining the tumor, it can be a really important way of not only diagnosing the tumor, but also thinking about therapeutic and indeed preventative approaches to managing this particular malignancy. So what often happens is the tumor, as you can see on the right-hand side, spreads from the back of the nose to the lymph nodes in the neck. And often people present with very late metastatic cancer. And that's a big issue for how we screen. But what we do know is that everybody that has NPC, all those people have elevated antibodies and indeed elevated levels of circulating DNA in their blood, that the tumor is consistently EBV positive with a restricted pattern of gene expression. We know that you can particularly find EBV in high-grade pre-invasive lesions, but not low-grade, something I'll come onto at the moment. So many of us are continuing to study this interesting tumor, mainly from a therapeutic perspective. So here's a nice picture demonstrating one of the key criteria for associating infection with a cancer here. You can see EBV in the tumor cells. Top left panel on the non-polyadenylated RNAs, and you can see staining for EBNA1, for LMP1, and for LMP2A. And what we know about these proteins increasingly is that they're playing an important role in the maintenance as well as the development of the tumor. And so we can study these different things. We know that, for instance, I said earlier, that the LMP1 and LMP2 molecules are important in cell signaling, and they affect cell signaling in the cells. And so there's lots of studies, including some recent work that we've contributed to on the way that NF-kappa B pathway is so important in nasopharyngeal carcinoma. The EBNA1 is there, and that EBNA1 not only maintains the viral genome, but has some effect on gene transcription and indeed in DNA damage. The BART microRNAs also affect many processes, including apoptosis. Then we have the EBAs that affect the innate immune response. And something that's a bit mysterious is a secreted protein that the virus makes called BARF1, which seems to interfere with macrophage development. So these are all interesting pathways that we are studying. And the idea is if you can identify and target some of these signaling pathways, these might add to the therapeutic approaches to managing this cancer. But in terms of diagnostics, this tumor is a real paradigm, because circulating DNA is now a very important way of diagnosing cancer and is being used for many different tumors. But again, the gold standard for this is EBV, because as the nasopharyngeal carcinoma cells grow, like any tumor, the tumor cells turn over. And as they turn over and die, they release into the bloodstream DNA. So that's very easy to measure. You can see some early studies that showed if you take a bunch of NPC patients, this is just a cross-sectional study, just compared to controls, you measure using quantitative PCR DNA loads in the blood, you see this range, but much higher than in the controls. This is also something that responds to therapy. So if you are able to diagnose NPC early, when it's still restricted to the nasopharynx, radiotherapy targeted to that bit of the nose works really well. And you can see that the levels of DNA fall drastically after radiotherapy treatment. But more importantly, you can also use this approach to look at prognosis in individuals. So you can start to measure these levels and see that whilst they fall after radiotherapy and the patient goes into complete remission, if you look at the top left here, you can see that as metastases occur, so the levels of DNA in the blood increase. In other words, you can use this as a marker of prognosis to guide future therapy, and you can often see these rises in circulating EBV DNA before you can easily detect the recurrence. And then taking it to another extreme, a paper published a few years ago now demonstrated that you can do population analysis, surveys, screening for nasopharyngeal carcinoma by measuring EBV DNA. This is a really exciting approach, and I'll leave you to look at that in a bit more detail. What we're left with then is an interesting scenario, like other cancers. You've got early stages and cancer development as the cancer spreads, going through these different pre-malignant stages. And what we believe with MPC actually is that the virus comes in quite late, because normally what the virus would do is it infects a differentiating epithelium, it would replicate. But in this situation where MPC develops, the virus can infect cells that have already undergone genetic changes, probably induced by environmental factors. Those changes prevent the virus from replicating, and the virus goes into this latent stage and then contributes to cancer that way. So it's quite a different process, but nevertheless, the virus is intimately involved in that process. And what we're trying to do now is understand more about how the virus is associated with metastasis and what we can do to prevent that. Different therapeutic approaches. I mentioned earlier the use of adoptive T cells. Well, they can be used in other cancers. The problem in other EBV-associated cancers is that many of the more immunogenic proteins, the antigenic proteins, are not expressed in some of these tumors because the virus downregulates many of its genes. So that can be a problem, but there are clinical trials going on now looking at EBV-specific T cells as an adoptive therapy for MPC. The idea of therapeutic vaccination is quite interesting, and there are trials where genes have been taken from the virus that we know are highly expressed in MPC, and then introduced into a virus vector. In this instance, vaccinia. Phase 2 trials of this going on now in Hong Kong as a therapeutic vaccine for patients with metastatic disease. There are now trials, again, just starting recently, going all the way back and thinking about how could you prevent people getting infected with EBV in the first place. Complicated because we all get infected very early in life, but there's a new trial recently started in America to look at the possibility of a prophylactic vaccine. But also there are new and exciting approaches to therapy. So using small molecules to block some of the proteins. One of the key proteins of EBV is the EBNA protein that binds to and maintains the viral genome. It's a very nice work from Troy Messick and Paul Lieberman. They've developed a small molecule that can block the ability of the EBV, of the EBNA1 protein to bind to EBV DNA. And that's hopefully going to go into clinical trials very soon. So what we've got here is a virus that is common in all of us, but through a variety of mechanisms can lead to different tumors within the lymphocyte lineage, but also acts in a slightly different way to contribute to both actually nasopharyngeal carcinoma and a proportion of gastric cancers. Now, looking at the time, I was going to go on and talk a little bit about human papillomavirus, but I think you've all got copies of the slides, so you can look at them and go through them yourself. But just very quickly, it's worth just mentioning the association of papillomavirus infection with cervical cancer and other tumors. This is a large family of very small DNA viruses, and they behave very differently from EBV. They're a very common infection, but they're very small. Their genomes are just about 8 kilobases, so very, very small, and they belong to the polyomavirus family. But they are associated with certain types. There are more than 100 different types, but certain types are associated with cervical cancer. So I'm going to skip a few slides because that's just and just show you a couple of important points about this. One important point is that whilst this is a virus that's associated with anal genital cancer predominantly, over the years, it's the association of virus infection with oropharyngeal cancer has been increasing, particularly in the West. If you look at the bottom here, you'll see that this is something that is on the increase quite significantly in Western communities in particular, where HPV-associated oral cancer is becoming much, much more common. And that's something that, again, is quite interesting, and why that's happening and what we do, we can discuss that. Because these virus genomes are quite small, in a way they're easier to study. What we know is these red proteins are the transforming proteins that seem to be really important in driving proliferation of epithelial cells. And then the other proteins are associated with the control of the genes transcription and DNA replication. What's interesting is there are two structural proteins, L1 and L2, that wrap up the virus in this protein coat. And those coat proteins are very important for vaccination. And that's what I want to finish with in a minute. Just a little bit about what the main transforming proteins do. The E7 protein is really interesting and worth looking at because it interferes with the function of retinoblastoma and it inhibits it releases the inhibition induced by RB. And what that does is allow the virus to replicate in post-mitotic cells. And then the E6 protein is also particularly interesting because it targets p53 for degradation. So what this virus can do is overcome cell arrest, post-mitotic cell arrest by E7, and it can overcome p53 control by taking out p53. So rather than mutating p53, a common feature of many cancers, E6 makes p53 disappear. It induces its degradation. And that means if you look in HPV-associated cancers, particularly in the cervix, you hardly ever find p53 mutation because you don't need that because the E6 protein is there. So looking at the time, I'm going to skip over these slides. You can look at them at your leisure and just come on to what I think is really important. The important reason for mentioning HPV is that quite excitingly, this L1 and L2 protein that forms the capsid can do that in the absence of the DNA. So if you just produce the L1 and L2 protein, you get virus-like particles, VLPs, forming. And this is the basis for the vaccines that have been rolled out over the last few years. I was very excited to be involved in with GSK and the development of their papillomavirus vaccine. And what the idea here is that you vaccinate individuals before they're sexually active, because HPV virus is spread by sexual intercourse. And if you vaccinate young women before they're sexually active, this should prevent cancer development. And it's exciting that you can use this VLP approach. And to demonstrate the fact that that actually works, here's some recent data published in New England Journal of Medicine, looking back at the cumulative incidence of cervical cancer per 100,000 people. And what you can see looking at these curves in the unvaccinated population, here's the age here, you can see the steep rise in cervical cancer. If you look here at the green dotted line at the bottom, that's youngsters that were vaccinated before the age of 17. You almost completely block the development of cervical cancer with this vaccine. And here in between are individuals vaccinated between the ages of 17 and 30. And of course, those individuals, some of them would have already been exposed to papillomavirus infection before they've been vaccinated. So that's problematic, and that's why you get this reduction in the incidence of cervical cancer, but it's intermediate. So studying this virus has been really interesting because it tells us a lot about the development of cancer, but also how exciting it is to be able to develop what is now a vaccine that's used across the world. And these are just different aspects of our understanding. What I haven't had time to mention is the point that this virus is a bit different from EBV in that to persist, it actually integrates its viral genome. So this is a different type of approach, and that can be quite dangerous. That point of viral integration can lead to the development of cervical cancer. HPV screening, particularly in the UK now, is driven by PCR testing. So primary screening is to see if the virus is there, because it's so important in the etiology of cervical cancer that if it's not there in the cervix when you take a cytological smear, then you know the individual is very, very, very unlikely to develop cervical cancer. And obviously there's an issue about the vaccines. Now, one of the big reasons for studying virus-associated cancer is that we've got a handle on one significant etiological factor, the virus itself. And that's something we don't often have for other cancers. Sometimes it's very difficult to find a key factor. But when you have a key factor and if you study it, it teaches you a lot about cancer generally. So it teaches us a lot about oncogenic mechanisms and how that works. And now in particular, when we're thinking about tumor microenvironment, neovascularization, studying these tumors really helps to unpick and dissect that, particularly I think around immune surveillance. I don't know if I've got immune surveillance there. I've got it twice, but evasion of immune surveillance is really important in all cancers, but studying the virus-associated cancers and how they develop away from the body's immune system is allowing us to develop and think about new ways of treating all cancers. And so that's where sort of therapeutic approaches are really important. And I just wanted to finish with a slide that shows a timeline. And the timeline is quite important because in 300,000 BC, a skull has been identified from that time that looked like it probably had Burkitt lymphoma, the tumor associated with Epstein-Barr virus. But if you go through all these key points in terms of our understanding of the impact of environmental factors on cancer, you can see that it's dominated in the last century by viruses. And we still believe, actually, there are going to be many other infections associated with the development that are yet of cancer that are yet to be discovered. And they will allow us not only to understand the cancer process in more detail, but to develop new ways of diagnosing and to treating cancer, not only relevant to the infection-associated cancer itself, but to all cancer. We've learned so many lessons from studying viruses and cancer, and now we're in an age, I think, where we're applying that to new therapies and new ways of diagnosing. And I'll finish there. Thank you.
Yeah, thank you, Lawrence. Yeah, I saw in the chat box there are several questions. Yeah, I will read question. The first question is from Ibu Leli Yuniarti. Yeah, she asked about the incidence of NPC in Central Java, Indonesia is high and often associated. Oh. Sorry. I start with the last question. The incidence of NPC in Central Java is high and often associated with the habit of consuming salted fish. Is it easier for EBV to causing chronic infection to people who often eat salted fish?
This has been a very this has been a very controversial issue. What is is there a role for salted fish? We believe there is, but what is that role? We know that salted fish contain volatile nitrosamines, and there are a couple of possibilities, and they might not be mutually exclusive. I mean, one is that given the fact that EBV can replicate in epithelial cells in our noses and throats, if you eat salted fish, it may be that the salted fish, the nitrosamines induce more virus replication and just increase the chances of NPC developing. I prefer the idea, actually, but we have very little data to support this, that one possible contribution of salted fish and volatile nitrosamines could be common changes that we see, genetic changes. So we see loss of chromosome 3p, 9p as a very common change in nasopharyngeal carcinoma, and a change that we believe occurs before EBV infects. So one possibility is that it's not about inducing the virus to replicate. What it might be doing is inducing genetic changes that prevent epithelial cells from differentiating. And we believe that that block in differentiation may mean that when the virus gets in, it can't use the processes of epithelial cell differentiation to replicate. So it forces the virus into this abortive approach, in this latent gene expression that might contribute to NPC. So we don't know, and there's still a lot of work going on trying to understand the specifics of that. But it is quite interesting to try and explore the interaction of EBV with salted fish.
Okay, bagaimana Ibu Leli?
Terima kasih, Bu.
Okay, yeah. So now we go to the first question from Yaya. For NPC or lymphoma, does virus play a role in severity or of the disease compared to non-viral related?
I think thank you, Yaya, for a very complicated and difficult question. I think well, for NPC it's difficult to compare. I mean, what's interesting actually, of course, is that we can't compare EBV positive with EBV negative NPC as we can with HPV positive versus HPV negative oral cancer. So we've got no idea about the degree to which the virus affects the severity of NPC. For lymphoma, it's a different situation, and it seems to depend very much on the type of tumor, because the virus is associated with a range of different lymphomas. If you look at Hodgkin's lymphoma, for instance, you often find EBV in more severe forms of Hodgkin's lymphoma, particularly the mixed cellularity form of that particular tumor. And you know, this is a difficult question. So I think we just don't know about the role it plays in the severity of the disease at all, and we don't have any data to suggest differences other than looking at gastric cancer. That's quite interesting, because in gastric cancer we can look at EBV positive versus EBV negative gastric cancer, and actually in that situation, if you have EBV positive gastric cancer, you do much better than if you have EBV negative. And the reason we believe that that's occurring is something we haven't had time to discuss today, which is the way that EBV contributes to cancer often involves epigenetic mechanisms. And epigenetic mechanisms are reversible. And if you look, as people have done, using genome sequencing of gastric cancers, you find that the EBV-associated cancers in the stomach have many, many more epigenetic changes. And by definition, because those changes aren't in the DNA, they're not somatic, they do tend to be more reversible. And indeed, they can be reversed with some drugs. So it's possible that because EBV in that context is driving a more epigenetic phenotype, it's more reversible and less severe. But we don't know that for NPC.
Okay, yeah. So how's Yaya?
Thank you, Lawrence.
Okay, and the next question is from the not from this list, but from other audience. In cervical cancer, having multiple partners is a risk factor. Does HPV come from one partner, or does the virus also come from the other partner? Do natural killer cells play a role in eliminating HPV in the treatment of cervical cancer?
Right, so we've known for a long time from the epidemiology that cervical cancer is associated with multiple sexual partners, and the obvious idea there is that it just increases both the risk of getting infected with HPV in the first place, and possibly getting more infection as well. There's a complicated and sensitive issue, which is to do with the way the virus infects and the abrasions in the cervix that might contribute to increased infectivity. But we've known for years that a major factor, and it's actually something we studied ourselves many years ago in epidemiological studies, is number of sexual partners, and we just assume that's just about the risk of getting infected and reinfected with HPV. NK cells are really interesting because, of course, we don't know that they play a significant part in eliminating HPV. There's been a lot of work obviously done on the T cell response to papillomavirus, and indeed some attempts to use T cell therapy, which haven't been overly successful. I think NK cells are interesting because they're not as reliant, obviously, on the MHC expression. And one of the big problems with all cancers, including cervical cancer, it can often be the downregulation of MHC. But I'm not aware of any, I mean, there are loads of studies on this, but I'm not aware of any specific data that particularly implicates NK cells in eliminating HPV.
Okay, yeah. And then we go back to the question number 2 from Nanny. Yeah, what makes some virus able to induce cancer while other can't? What do you think of the SARS-CoV-2?
Yeah, so I think that is a really interesting question, because I think a lot of this is around the way the viruses behave. What we're seeing here is that viruses associated with cancer are often viruses that are persistent infections. So these are viruses that have evolved to infect us and stay with us for a long time, stay with us for always. Herpes viruses like EBV, Kaposi sarcoma virus, polyoma viruses, Merkel cell, papilloma virus. These are all viruses that when you get infected, the infection stays with you for life. It's not an acute infection, whereas viruses like SARS-CoV-2 or influenza virus are acute infections. They replicate in our bodies, they grow, produce themselves, produce acute symptoms, but then they become eradicated. They don't stay in your body forever. So I think the big difference is the way the persistent viruses work. That doesn't mean that some of these other viruses can't contribute in other ways to cancer, perhaps, but I think it's more to do with persistence and the mechanisms that these viruses have developed to persist that can sometimes go wrong and contribute to cancer. So I think the big difference here is viruses that are persistent and stay in our bodies forever once we're infected, versus acute virus infections that after they have grown, replicated, our immune system usually completely eradicates them.
Okay. And then the last question is from me. The infection in cancer pathogenesis should occur chronically, but how about in juvenile NPC? How is the involvement of EBV in the pathogenesis? And is it similar with adult? And also, which EBV genome most involved in juvenile NPC?
I think juvenile NPC has been understudied. It's really very interesting. We see it in certain parts of the world, particularly in North Africa. And if you look at it, it does appear to be slightly different in terms of EBV gene expression. So we know, for instance, that the oncogenic LMP1 protein is more consistently overexpressed in juvenile NPC. But what surprises me is there haven't been enough studies, or hardly any studies at all, on the genomic changes in juvenile NPC, on the possibility that there are different types of EBV. One thing we haven't discussed, and it's great that you've raised that, Dowie, is the fact that, as we're hearing all the time about SARS-CoV-2, of course, there are variants of EBV around the world, and it's been difficult for a variety of technical reasons to sequence the genome until now. We have much better methods now, but what we're learning is that there is some sequence variation, some strain variation in EBV that could contribute to adult NPC, and we've seen some recent publications identifying regions of the EBV genome where there are polymorphisms that might contribute in some way. But I haven't seen any studies in juvenile NPC, and I think it'd be really interesting to look both at the underlying genetics, the genotype and host cell genotype of the changes in juvenile NPC, and also whether EBV is slightly different in those tumors. That hasn't been studied, so I think it's something really interesting for the future. As with all these studies, it's sometimes just difficult to get hold of samples, but I think juvenile NPC is something we should really try and focus on a bit more, because if we've learned anything from other cancers, is when you study cancers in youngsters, you learn a lot more about how cancer is developing, because obviously in children, often a number of the changes associated with cancer have happened very rapidly. We know with leukemia, sometimes these changes occur in utero, actually. And I think it will be really interesting to try and focus a bit more on juvenile NPC.
Okay, thank you, Lawrence. Yeah, that's all the question, but may I have one more question? Yeah, because sometimes, yeah, for the infection-related cancer, now many people also developing the therapy to the virus. We can prevent the cancer by preventing the infection and etc. How is the potential of therapy if it's already happened? How is the potential of using therapy using a viral component in combating the cancer?
I mean, targeting the virus.
In general, yeah.
Yeah, it's interesting, isn't it? Because really, we haven't seen much of that in virus-associated cancers at all. Targeting, because on the one hand, because these viruses tend to target common pathways, then some of the signaling inhibitors that are being used and tested are quite interesting. Although, as we both know, for NPC, these haven't worked very well at all. So, for a good example would be the EGF receptor pathway. They're really good tyrosine kinase inhibitors that have been used for other cancers like lung cancer. We all thought, because we know that LMP1 and other EBV associated molecules can affect that signaling pathway, we all thought that targeting the EGF receptor would be useful, but in all the clinical trials that hasn't proved useful at all. And one of the problems, of course, is that we can treat primary NPC with radiotherapy. So all the time what we're trying to do is develop better approaches to metastatic NPC, and it's worth just mentioning, of course, that conventional chemotherapy doesn't work very well at all for metastatic NPC. So I think what it is exciting to, and even now looking at, for instance, immune checkpoint inhibitors, we thought perhaps for virus-associated malignancies, because the way they work and because the way that the immune system sees viruses, they would work really well, but they don't. They don't work any better than non-virally associated cancers, actually. For the majority of cancer, you get about a 20% response rate. So I think targeting EBV more specifically might be interesting. So particularly with the EBNA1 blocker, because EBNA1 is so important in maintaining the EBV genome. that if you can get, if you can stop that happening and combine that with chemotherapy, my view is that, you know, there's an expression we use in English, which is you shouldn't throw the baby out with the bathwater, which means, you know, for 50 years, we've learned a lot about combination chemotherapy. Combinations of drugs targeting different pathways are really important in cancer. So I don't think a single approach like the EBNA1 inhibitor will work, but I think combining the EBNA1 inhibitor with other chemotherapy or with immunotherapy will be very effective. And I think working on that, and I'm particularly interested, as you know, that in the BAF1 protein that's secreted by MPC, and I think that could be a really interesting target for therapy as well, because that may well influence the tumor microenvironment. So I think we've got a long way to go, but I do believe there is a future in targeting the virus pathway, the virus-induced cell signaling pathways and other pathways as therapies.
Okay, thank you very much, Lawrence, for the fruitful discussion. And now we are at the end of the session for today. But before that, please don't leave. We will have a photo session with all of the participants. Oke, silahkan fotografer. Sebelum kemudian nanti saya tutup.
Bapak Ibu, silahkan dibuka kameranya ke videonya untuk foto bersama.
Lawrence malah ditutup.
Oh iya, pada Awi?
Lawrence, would you please just put your camera on?
Yes, because we will have a...
Thank you.
Mbak Rika?
Yeah, since there are 8 pages, so please be patient. Okay, I will start with page 1. Page. Okay, please keep your smile for a while.
Would you please inform us in which page are we now?
Okay. We are in page 6. So.
Okay.
1 more pages. Okay, thank you very much all.
Okay, thank you very much, Lawrence. Ya, dan untuk Bapak Ibu peserta, jangan lupa untuk mengisi evaluasi yang sudah disampaikan tautannya di WA Group. Lalu kemudian seperti yang lalu, Anda bisa nanti membuka elok dan mengakses kuis yang mungkin mulai minggu ya, Bu Neni ya?
Ya, untuk yang akan mulai kita buka hari Minggu.
Oke, akan mulai kita buka hari Minggu. Kemudian untuk minggu depan sesi akan dimulai pagi, yaitu jam 8.30 pagi seperti yang lalu. Oke, terima kasih untuk Bapak Ibu atas perhatiannya. Ya, sampai dengan sore hari ini, semoga apa yang kita dapatkan hari ini dapat bermanfaat untuk kita semua. Terima kasih. Selamat sore. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam warahmatullahi wabarakatuh. Matur nuwun, Bu.
Waalaikumsalam.